assess all these things, these assessments are done, right? So, number one, it is the Wechsler's intelligence scale. So, it is a very, very important scale to, it is also known as or abbreviated as WAIS 3 for the adult intelligence scale, and WISC 3 for the children. Okay, so there is a particular age limit also. So this is the first version of the intelligence test, and it was known as the Wechsler's Bellevue intelligence scale. So this is a very important scale of intelligence test, and it was published in 1939 by David Wechsler. So it is named after his name, that is Wechsler, and to measure mainly the intellectual performance of adults, right? So these scales have been subsequently been revised various times for a particular age group, like for adults, for early childhood, for late childhood, for adolescents, like this. So the Wechsler Adult Intelligence Scale 3 is administered on adults, and this intelligence scale is also for the children, and mostly it is important from the age group 6 to 16. That means in 6 years a child goes to class 1, for example, and 16 years means that child is in his or her adolescent period, right? So this is the two important Wechsler's intelligence performance test. Now the next one is the Halstead-Rating neuropsychological test battery. Now this is another important neuropsychological test battery, because it is often used for training purposes and also for the clinical psychologists to assess if there is any behavioral issues or problems. So the battery is designed to assess all major, number one, the cognitive, the sensory, the expressive, and the motor functions. So it was said by Re-10 in 1986. And this is known as test battery because there are various questions, and the whole the format of the questions, it is known as the test battery, right? And this test battery is found to be reliable and valid for both the sections of the group, that means the younger as well as the older age group. Now the next one is the Luria, Nebraska neuropsychological battery. It is another important battery. Obviously, it is a standardized battery, and it was first came into existence in 1986 by some neuropsychological tests, where it is very important for diagnosing any treatment of any accident or any brain damage, or if any portion of the brain, that means the cerebellum or the cerebrum or pons, thalamus, etc., if any portion of the brain is not functioning in a proper way, then this type of test battery are mostly used. Now, the test battery is useful for the adolescents, the adults, and also for the older adults. That means above 60, right? So there is separate versions for these age groups, and this major areas covered all the assessment part, like the motor activity, the recognition, then the memory, then arithmetical ability, maybe the language or the spelling, then the maybe the communication skill, etc. So the next test is the Ray Ostrich complex figure test. So it is the number 4 neuropsychological assessment test. Now the test was designed by Ray in 1941, and it is used to assess only the visual perception and the visual memory in brain damaged individual. So only the visual section of the person is assessed. That means the visual perception and the visual memory. So if there is any damage in this portion of the brain, then this type of use can assess that yes, this part is the problem area and the individual is facing this type of problem due to the damage of this portion. So it is one of the most used tools to assess the constructional abilities and is also used for the non-verbal memory test. Why for non-verbal memory test? Because it is mostly a test to assess the visual perception. That means an individual who cannot speak, that person will also be able to do this type of tests. So this is a figure test, a complex figure test, and you have to assess it. You have to just like a puzzle box. So a person who cannot speak also can easily solve this type of test. Okay. So the next one is the Dellis-Kaplan executive function system. So it is another most important neuropsychological assessment for the executive functions. Now, what are the executive functions? Like the cognitive functions, like thinking, then like perception, or any problem solving, any critical thinking, any abstract thinking, then maybe some creativity, like an artist. Okay, so these type of mainly the cognitive part. Okay, so it may be administered individually or on a group. So this is a very important part, that this test is also administered in individually as well as in group. And it is a standardized measure and to assess the executive function in children and adults between the age range 8 to 89. See, the range is a huge one. See, from the late childhood to the 89 means the mostly the old age. So this is another important neuropsychological assessment test. Then the digit span test. Now, what is digit span? It is another important test to assess the working memory. So, what is working memory? It is a short-term memory. That means regularly we are hearing something, we are seeing something, and this first goes to our short-term memory, and through rehearsal, and this short-term memory is known as the working memory. And that person, the aim of the test mainly is to assess or to measure an individual's short-term memory, the capacity or the ability of this short-term memory, and to assess any impairment of the waking memory. Now, what is waking memory? Now, waking memory means when we are in a conscious situation, we are hearing something, and that goes to our short-term memory unit or the working memory. So this is the another important neuropsychological test. Then the Wisconsin card sorting test. It is from the name you can just know about that. There are some cards, and you have to arrange it in some ascending or descending order. There are some instructions given there, and it is also known as the set shifting test. Set SET, set shifting test, and it is is used to assess the frontal, mainly the frontal lobe dysfunction. So when I will teach you the brain, then you will be able to know that which portion of the brain is the frontal lobe. So the the dysfunction of this frontal lobe. So mainly this portion is assessed by this type of set shifting test or Wisconsin card sorting test, and this is very important test for mainly the patients who are having any brain injury or some psychotic disorders like the schizophrenia or another mental illness like the paranoia or some psychosis, etc. And the participant has to sort, there are 64 cards to match either with color or maybe some numbers or maybe some particular figures are there. You have to arrange the figures accordingly, etc. And here the participant is not explicitly told the rules or the regulation of this test. So you may give the feedback of that test, but there is no some instructions given to the patient or to the subject. Okay, so just you can give a feedback that previously this person has done this type of card sorting. So you can just tell like this, but there are no some fixed information or regulation or rules given to the patient just before the test. Then the dichotic listening test. Now, di means two and otic means ear. So, dichotic means 2-ear test, right? So, this is an non-invasive test and initially used to investigate some selective attention, but later used to assess the hemispheric lateralization of language ability. That means if a person is blind, he cannot see anything, then this type of dichotic listening test is very important for his or her to assess mainly the language ability and the standard dichotic listening task. That means how he or she is hearing any object or any sound and how he or she is responding. So this is another important dichotic listening test, another important test of this neuropsychological assessment. Now, after this slide, I will show you the what are the research ethics in biopsychology. Now, there are some fixed ethical guidelines that we have to follow. So what are they? Like the American Psychological Associations, and in India also there are some guidelines given by the Indian Council of Medical Research, and there are some instructions or guidance to the researcher while conducting this research with some human and the animal participants. Now there are various ethical issues that need to be taken care of while doing this research in biopsychology. So if you want to investigate a problem area of a particular portion of a brain, then obviously you have to do some of the methods that you have seen in the previous slide. So what are the ethics that we should follow? So for such studies, as well as for another studies where there is harm to the person or where survival is at risk, then animals are preferred for investigations like maybe rats, guinea pig, etc. Okay, cat, dog. Now when human participants are selected, then the consent is taken for the participant and the doctor or doctors and treating him or her or therapist and the caregivers. So whenever it is felt that the experiment is disturbing the participant or negatively influencing his or her symptoms or maybe aggravating any medical or psychological condition, then the experiment is immediately discontinued or stopped. Right? So these are some of the ethics in biopsychology. Now, today I will start from the next unit, that is the most important chapter of this 102, that is the neurons and the nerve impulse. So, I will just share the screen, just a minute. Okay, so the first slide is there is a explanation about the structure of a neuron. So, what is a neuron? First of all, that portion is to be firstly the definition part mainly. That means all organisms have cells, right? So all organs within the human body have also cells. Like there are some, say, for example, brain cell, for liver cell, kidney cells, etc. Now, our behavior, our actions, our thoughts, our movements, our interactions, anything, these are controlled by the cells, the chemicals and the organs, right? So, nervous system is one such complex arrangement of cells, and that cells of nervous system are the neurons. Okay, so William and Harrop in 1988, they said that approximately there are 100 billion neurons in an adult human brain, and there are 12 to 15 billion neurons in cerebral cortex and some associated areas. There are some other research works or findings like 70 billion neurons in cerebellum and nearly 1 billion neurons in spinal cord. Now, these neurons are also of varied shapes and sizes. That means in a particular organ, maybe that part consists of some small neurons and another part may consist of some long or large neurons. So, these neurons are also protected by some cells which are known as the glial cells. It always protects the neuron. It surrounds the neuron. Now, let us see that what are the structures of a neuron. Now, the cells of the nervous system that receive and send messages within that system are known as the neurons. This is the definition part of neuron. So, you might get that what is neuron. Now, I will show you the next slide, that is the diagram. See the structure of neuron. So, soma, that is the cell body, that is the main part. Then the branch of a tree-like sections, that is the dendrites, and the axon inside, which is covered by a sheet, that is the myelin sheet, and the direction of the messages, see, the arrows. These are the directions of the messages, and this neuron is connected with another neuron by some terminal buttons. Okay, so this is the main structure of a neuron, where we can see that the soma or the cell body and the axon, the terminal buttons, and the main fluid present in the soma, that is the plasma membrane, the branch like trees section are the dendrites, the myelin sheath, and the primary cells that consist are the neuroglia and the glial cells and the sconce cells. So these are the main structures of a neuron. Now, what are these glial cells, or it is also known as glia? Now, these neurons are present in large numbers in brain, and there are other some primary cells also that always protect or supports this neuron, just like a covering. So these cells like the neuroglia or the glial cells or glia, they affect the thinking, the learning, the memory, the perception, that means mainly the cognitive part, and help in maintaining a state of homeostasis of the nervous system. Now, there are some neurodevelopmental disorders. These are like the autism or ASD, autism spectrum disorder, or some degenerative disorder like Alzheimer's disorder, or some psychiatric disorders like schizophrenia, depressive disorder, etc. These are due to this glial cells problem area, and there are 4 types of main glial cells, that are the oligodendrocytes, number 1, number 2, the sconce cells, number 3, the microglia, and number 4, the astrocytes. Now, what is this oligodendrocytes? These produce the myelin for the neurons in the brain and as well as in the spinal cord. So, as you have seen, the central nervous system is mainly divided into brain and spinal cord. So, these oligodendrocytes, they produce the myelin for the neurons in the brain and the spinal cord. And the sconce cells, they produce the myelin sheath around the neurons of the body. That means the peripheral nervous system, or the PNS. Now, there are another important part, that is the nodes of Renvier. What is that? Mainly, there are some sections like the myelin, it protects the axons. It is a membrane-like substance, and this such nerves it also known as some myelinated fibers, and there are certain gaps between them. Okay, so these gaps are known as the nodes of Renvier. See, there are certain gaps here, so these gaps are known as these nodes of Renvier. Now then, the next slide is about the classification and types of neurons. Now, on the structural basis, there are some classifications like the pseudo-unipolar, the bipolar, and multipolar. So, bi means 2, that you know. Unipolar means 1. One polarization is present here, that is the unipolar neuron. So, on the structural basis, you can divide neuron into these main 3 parts. Okay, so the classifications are the types of neurons, that is the pseudo unipolar, or it is also known as only unipolar neurons, because it has one axon that separates from the soma and branches into two sections. Okay. So these neurons are involved in only the sensory functions. Now, what are the sensory functions? That means the sensations are eye, ear, nose, touch, okay, taste. So these sensory functions only.
Ma'am, and the sixth
Now,
one is six skin.
yes?
Ma'am, the sixth one is skin, right?
Yes, yes. Skin also. So these neurons are involved in sensory functions only, and they send the impulses received from the environment to the central nervous system. That means the brain and the spinal cord. You all know that central nervous system is divided into the brain and the spinal cord. So this is about the pseudo unipolar neuron. Then comes the bipolar neurons. This is also another neurons for the, or it is also known as the sensory neurons. That is, they have they have one axon and one dendrite, which is profusely branched like a tree. So the dendrite is placed at the opposite end of the soma, and they are very few and are the kinds that are present in the retina of the eye, the ear, inner ear, then the olfactory part, that means the nose. So these all are the sensory functions. Then comes the multipolar neuron. Multi means more than two. So the multipolar neurons have only one axon, but there are several dendrites, right? So they are the most commonly found in the brain and the spinal cord region. Now they are further classified into two Golgi types. That means Golgi type 1 and Golgi type 2. Now Golgi type 1 neurons, they have very long axons and only few branches to carry the information of mostly the motor nerves. That means for the movement, muscular nerves. Okay. And the Golgi type 2 neurons are the short, they have shorter axons and the branch repeatedly. So these neurons act mostly locally to nearby areas. So these are all about the structural basis of the classification of neuron. Now, classification on the basis of functions. Now, functions means how they work. What are the functions of neurons? So, according to the functions, we can divide neurons into afferent neurons, efferent neurons, and interneurons. Now, what is the afferent neuron? Afferent neurons are the sensory neurons again that carry the nerve impulse to the CNS or towards the brain and the spinal cord. Now they are affected by the small changes in the environment or in the damaged area of the brain. So the efferent neurons, the next part or the next classification, that is the efferent neuron, it carries the impulses away from the brain or the spinal cord to the muscular or the muscle region and the glands. There are various glands present in our body. So from the brain and spinal cord to these muscles and glands. So they are also known as, the other name of this type of efferent neurons is also motor neurons. Now there are some interneurons also present. that lies within the CNS or the central nervous system, or which connects these regions, and they carry some information from the afferent neurons towards the efferent neurons. Now, then, so these are the three classifications of neurons according to the functional basis. Now, the next slide is about what are the main functions of neuron. Now, functions of neuron, first and foremost function is the reflex action. Now, what is a reflex action? Like, for example, blinking of eyes, okay, or any complex problem solving, or any communication between some sensory and motor neurons, etc. So, for example, if we are able to see an object, it is possible because the receptors in the eye communicate this to the brain, that yes, there is a danger, or there is a fire in front of my hand. So, there is a chance of burning, so I have to move my hand immediately. So, these are the reflex action. Now, the next important function is the problem solving, or to communication between the sensory and the motor neuron. So, by seeing any danger, immediately the message or the communication goes to the brain that yes, there is a problem here, or there is a risk situation. So, we have to take an immediate action for the risk situation. Then comes the intricate network of connection between the CNS, that is the brain, spinal cord, and with different parts of the body. That means the limbs, the hands, then the circulatory system, maybe the digestion, maybe the excretory system, etc. Now, another important function is the genetic as well as environmental factors that influence the development of the nervous system. So, these are the 4 important functions of brain from where you can get a very, it is a very important questions also for you, I am telling, because from the past few questions, you may find that what are the definition of the neuron, what are the types of or classifications of the neuron, and as well as what are the functions of neuron. Okay, so the next slide is about the neural conduction. Now, neural conduction means from the term conduction, it means to pass through, right? So, like for example, electricity. So, a neuron that is at rest, that is not firing a nerve impulse or a message, is actually electrically charged. So, there is a jelly-like solution or fluid inside the cell, and this fluid consists of some charged particles which are known as ions. Now, the ions may be of positive or may be negative. So, positively charged ions are known as cations, and negatively charged ions are known as anions. Now, some positively charged ions are like Na+, that is the sodium ion, then the calcium ion, then potassium or the kalium ion, and see, these are all cations, okay, because there is a plus sign. And chlorine, this is a negative ion, that is anion. Okay, so these are all about the ions. There are 2 types of ions, and The positively charged ions are that I told you: sodium, calcium, potassium and chloride, and there are relatively more positive charge next to the plasma membrane on the outside and relatively more negative charge on the inside of the membrane of the cell
Ma'am,
or
chlorine
the
is the,
neuron.
ma'am,
Yes,
ma'am, chlorine is the only anions.
yes, chlorine is a negatively charged ion. Okay.
Okay.
So this difference in this electrical charge across the membrane, it is known as the membrane potential. Now there are some resting membrane potential also, because when it is inactive, when it is in an inactive state, that means the neuron is in an inactive state, then this resting membrane potential takes place. So it displays the membrane potential or the resting potential of minus 70 millivolt. Millivolt means small letter M, and V, capital V, it denotes the volt. Okay, so this is known as the resting membrane potential. It is also abbreviated as the RMP. So it maintained with the help of some principles. Now, what are the principles? Like the diffusion, the electrostatic pressure, the ion channels, and the sodium potassium pump. So, in chemistry class, you have studied about this, all of this electrostatic pressure. What is the ion channel? That means the negative, positive always attracts positive, always attracts negative. Okay, so these are some of the resting membrane potential, or RMP, is maintained by following these principles like the diffusion, the electrostatic pressure, the ion channels, and the sodium potassium pump. Now, the nerve impulse is brief and it goes through the cell body to the axon to these terminal buttons. Now, let me show you this. See, there are some terminal buttons here. Have you seen? So, through these terminal buttons, these impulses or these nerve impulses brief, and it goes through the cell body to the axon to these terminal buttons. Now, now when there is a stimulus at the spike initiating this region, that reaches the threshold of excitation. Now it depolarizes the membrane. That means the plus, it attracts always the negative or the anions. So this leads the sodium ions to move into the cell with the help of some forces. Now these forces always follow some principles that I told you, like diffusion or via some electrostatic pressure. Okay, so this makes the inside of the membrane more positive with respect to the outside for a temporary period of time, from say about minus 70 millivolt to plus 50 millivolt. So this action potential, that is the electrical charge reversal at a particular point along the axon, it reaches its peak in about 1 millisecond. See, it is so fast. Okay, so no more sodium can enter the cell and potassium ions start leaving the cell. So this is the process of neural conduction. So this causes the membrane potential to restore itself to its resting state. Now the next slide is about the synaptic transmission. Now synapse, it is an important part of a neuron. So the information passes from one neuron to the another neuron through this section, that is the synapse. Now, so the action potential, it reaches the end of the axon, then it reaches the round-shaped terminal buttons, and this point where the terminal buttons from one neuron contacts with the dendrites of another neuron is known as the synaptic region. Now, the information, it passes at this junctures formed with the next neuron. So, there are some three places, or synapses occurred in three important places on the next neuron, mainly on the number 1 dendrites, number 2 soma or axons. So the synapses are known as the axon dendritic or axon axosomatic and axonic synapses. From the name, you can find out that these are the three places of the next neuron where the synapse occurs, and there are two kinds of synapses, like the electrical synapses and the chemical synapses. Now, the electrical synapses are very, very rare. So in this case, the information from the transmitting neuron is sent to the next neuron via these channels that come close to one another. And the chemical synapses, it involves the transfer of neurotransmitter from one neuron to the other. Okay, so these are the main. Now let me stop here so that you can ask me some questions. So I'm giving a few conclusion about this synaptic transmission. So synapse, the terminal buttons, then I told you about the axodendritic or axosomatic or axoaxonic synapse, and the types of synapse, mainly the electrical and the chemical synapse. And there is another important region of synapse, that is the synaptic cleft. Now what is that? There is a gap in between. So when the membrane of the cell that sends the information, presynaptic membrane, comes in contact with the membrane of the cell that is receiving the information, or the postsynaptic membrane, so there is a slight gap. So that gap is known as the synaptic cleft. Okay. So today up to this. Next day I will start from the structure of synapse. Okay. So if you have any question, please ask me. You can ask me. So, yes.
Ma'am,
Mhm.
ma'am, this is for only our classes. When I visit the RC, they told when the counseling is done online, we have to write this on our language and submit it on RC. It is not clear to me, ma'am.
Now, see, this type of problem you have to ask to the office. So, if you have any problems regarding this chapter or this unit, so you can ask.
Okay, ma'am.
So, if there is any question, you please ask me. Yes, next. Yes, Purbasha, you have raised your hand.
Hello.
You can ask me.
Hello, ma'am.
Yes. Hmm?
I want to know that class is only for today or regular?
You will get the dates. Okay.
Okay, ma'am. Thank you.
Mhm.
Ma'am, but the links of the other classes has not yet been sent to us. Only this about this class, only the link has been sent. But the other classes which you took in YouTube and all, I have seen that. So, but the other classes, 3 classes, they have scheduled and they have sent the scheduled timings, but they have not sent the link to us.
Oh, that you have to ask to the office. So that part I don't know. Yes. So, yes, next one. You can ask.
Yes, ma'am. When is your next class?
Maybe it is on Saturday. So you will get the date.
Ma'am,
See the yes.
ma'am,
Srija?
in the email it is written that let me see. Yes, the next class is on 21, then on 26 and 28. Will it be on same link only?
That is also a technical question. Sorry, dear. You have
Yes,
to ask
because
it.
yes, in the same link only it is provided all that date. That's why. Otherwise, we will receive, I guess.
I think you will receive if there is
Okay.
any other link. Okay. So don't worry
Okay.
about it. So if you have any question regarding this unit, you can please ask me. Yes, any one of you? Harita, do you have any question? Purbasha, you can ask me in any language. Yes,
Hello?
yes, Gopal. Proceed.
Ma'am, that class is being uploaded to YouTube.
Yes, in YouTube also you can see, you can watch.
Okay, ma'am. Thank you.
Yes, Purbasha, do you have any question? Harita?
নো ম্যাম, আমার ওটাই জিজ্ঞাসা করার ছিল যে আমি পরের ক্লাসটার জন্য লিংক কোথা থেকে পাবো, তো ওটা তো আমি জেনে নিলামই।
লিংক পেয়ে যাবে, চিন্তা করো না। যদি এই লিংকে না ঢুকতে পারো তাহলে তোমরা আরেকটা মেল পেয়ে যাবে অবশ্যই।
ওকে, থ্যাংক ইউ ম্যাম।
হুম। আচ্ছা। ঠিক আছে, তাহলে তোমরা কোয়েশ্চেন উনি তোমাদেরকে বলে দিয়েছেন, তোমরা নিশ্চয়ই বুঝে গেছো কিভাবে তোমরা ক্লাসটা দেখবে। ইয়েস, লক্ষ্মী, প্লিজ প্রসিড।
Ma'am, what is all or none law?
What is
All or none law.
okay? All or none law. That means where do you get it? In which portion? Yes, Lakshmi. Hello.
From the textbook.
Textbook of which unit? Unit 1 or 2?
Unit 2.
Unit 2.
Yes.
Where? Tell me. Today I have covered this portion.
Yes.
Where? Tell me the page number of your study material.
Page number 39.
39.
Just
Okay.
before the synaptic transmission.
Okay.
Ma'am, that's not yet covered.
Before the synaptic transmission. Yes.
Yes.
That part is not covered yet. That is hyperpolarized, right?
Okay.
Okay. So this portion I will start, as today there is no time. So I will describe this. That is the all or none phenomenon. This is an important phenomenon where the most important impulses will transmit, and that impulses which are not necessary or which are not emergency, it will restrict that. That is the all
It
or
is
none.
covered.
Yes.
This portion is given here. The action
Yes,
potential.
I know that. Yes, I know that. It is included in the neural conduction area, right? That is
Yes, ma'am.
before the questions. There are some questions like, what do you mean by membrane potentials? And just
Yes, ma'am.
before that, it is given. So this is the all or none phenomena that I told you. That is the impulses that goes through the axons, the terminal buttons, to the next neuron. So this information is sent down axons via there are some electrical impulses, which are known as action potentials, and all or none means all the potentials are those important potential, the impulses which are transmitted from one neuron to the next, which are emergency, which are very useful, so that portion will go to the next region, right? So this is the main meaning of this all or none phenomenon.
Thank you, ma'am.
So the next day I will start from this synaptic transmission. So please go through this section. If you have any queries, then you can ask me in the next class. Okay? Thank you, everybody.
Thank you, ma'am.
Thank you, ma'am.